Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Alright, so today we're going to learn chapter 1, cell divisions. Here is the objective for this chapter. Okay, you can see that a few objectives here. And then this is the outline for this chapter. We're going to learn on the concept of the division, the cell cycle itself, mitosis, and also the meiosis. Right. When we're talking about cell divisions, first, cell division is actually to produce a new cell, which is whether it's identical or non-identical. So cell division is a basic process of the cell itself for growth, for replacing the worn out cell, and obviously for the cell reproductions. Alright, so we're going to look at the concept of cell division. Here is the concept of cell division. The first and second theory being proposed by the Childens in 1983 and Schwann in 1989. Alright, so and then the third theory is come in the 1855, which is by Rudolf Virchow. Alright, there are two types of cell division. We have to bear in our mind. Cell division, they have two types with two different processes, and also we have the different product. Okay, the first one that will be a mitosis. Okay, so the mitosis itself, it will produce two daughter cell which identical, which is exactly the same like the parent's cell. Okay, whereas for the meiosis, they will produce four daughter cells which contain half number from the original or from the parents. Alright, followed by this cell division, there must be a karyokinesis and also the cytokine. There are differences between nuclear and cell division. For the nuclear division, they are similar for plant and animal. For cell division, they are different. Okay, nuclear division, they involve replication of chromosome. For cell division, they involve duplication of organelles. For daughter cell, they have similar and dissimilar for the mitosis. For the cell division, the daughter cell always similar. Alright, so nuclear division, the differences is they have a spindle formation. No spindle for the cell division. Alright, for the nuclear division, they're followed by cell division. And for cell division, they're always after the nuclear division. Alright, so what we have is the chromosome here. Okay, we have a chromosome. Human, we have 2n equals to 46. Alright, so this 46 chromosome will be in our each of our cells. So we have to remember human is made from a trillion of cells. Okay, so every single cell have this what we call the chromosome. This chromosome known as our genetic information that store everything for that human and unique by your own DNA. Okay, meaning that everybody have different DNA. Right? For example, this person, okay, they have their own color eyes, they have their own skin color, they have their own height, and also the weight. Everything will be determined in our DNA, or in other way around is actually being determined in our chromosome. So we're going to look at the basic term in basic genetic. Alright, first we call it a homolog chromosome. Is actually homo from the word homo meaning that same. So everything in this what we call the chromosome, there will be in a pair. Okay, so human have 46. Okay, but in the homolog chromosome, we call is a 23 homolog chromosome, meaning that homolog meaning that they are same. Alright, we're going to look at the chromatid. Chromatid meaning that this one. Alright, so chromatid is a half from the duplicate chromosome. So basically, they are, if they have two, we refer this is as a sister chromatid. Centromere is as here we have a joint at the kinetochore where joined by the sister chromatid and at the centromere. This is a pictures of chromosome number 1. Right, so this chromosome number 1, they have band, okay, band, light bands and the dark bands. Let's say we take one of these band here, right, which is known as IQ 21.2, right? So they consist of lots of what we call this genetic information. So we cut into this one. Okay, so we're going to look at this one, right? So actually this area consists of hundreds of information, or known as the information that control our appearance, our physical appearance, our chemicals, our hormones, and all the proteins synthesis. All right, so this is, we have lots here, right? So from only one plant. Okay, so human is made from a trillion of cells, which is identical to the gene. So there are contained is a chromosome, which is found in the pairs. So meaning that pair, one from one from mother and one from the father. All right. So these chromosomes, they are have the genes, which is, this is the location of the genes. Okay, which is control the physical and chemicals for this appearance for the humans. Alright, so we have a set of humans chromosome, which is, we have 46 chromosome, but let's say we want to talk about homolog chromosome. Okay, we only have 23 homolog chromosome, because when they are individual, known as a chromosome, when they are in a pair, okay, we call it as a homolog chromosome. Okay, so we have pair number 1, 2, 3, and so on till the last one, which is the sex chromosome. Whether the boy we have X, Y, for the girls they contain the X, X. Alright. Then we have a tetrad, okay, which is known as a 4 homolog chromatid or 2 homolog chromosome. We have a synapsis, a process of associate of the during prophase 1 of the mitosis. We do have a crossing over, whether it's a break or rejoin the homolog, which is non-sister chromatid during the prophase 1. I'm going to learn later. So we do have a chiasma, which is actually is a point where non-sister chromatid join and exchange between the chromosome chromatid occurs. Okay, so this is where the kinetochore, or known as a spindle fiber, attached to the chromosome. This is happen during the metaphase or early prophase in the meiosis or the mitosis process, where they have to hold the chromosome into the right place. Okay, we go to the cell cycle. Alright, inside for the cell cycle is actually a sequence of the event which the one cell divide for the next one. Duration of this depend on the type of cell and also the environment, such as temperature, oxygen, also the nutrient. Alright, I give you one of the example. Bacteria might divide every 20 minutes, meaning that we have, let's say we have classes for 1 hour. So the bacteria might divide for 3 times already. Okay, so why they divide so fast? Because they're quite small and they can form colonies of bacteria. Okay, cell lining of the epithelium in our small intestine. So they might divide for every 8 to 10 hours, not 24 hours. So meaning that every 8 to 10 hours, meaning that in a day, they will divide for twice. Alright, onion root every 20 hours, and surprisingly, the neuron, which is our brain cell, do not divide at all. Right, I will leave it to you and find why the neuron do not divide. Alright, so meaning that the cell cycle itself, they are depending on where the location of the cell itself and what is the task or what is the job for the cell itself. So they might affect to the rate of the cell cycle. Right, so I give you one of the example. Epithelial in the small intestine every 8 to 10 hours, but epithelial in our skin, for example, in our cheek cell, in our hand, and so on, they will divide for every 4 weeks or approximately 28 days. So the cell cycle, they are different from each of the cell, and most importantly, they are relying on the program having put in their chromosomes. Alright, So in the cell cycle, there are three main phases. Okay, they have the G1, S, and also G2. All right, they have the nuclear division, which is a mitosis and meiosis, and the cell division, or known as a cytokinesis. We have a pictures of the cell cycle. Right, one, the cell undergo what we call the cell divisions. So they'll undergo what we call interphase process. So please bear in our mind, so interphase is not a resting process. So actually they are very busy doing the duplication, replication of organelles and preparing for the showtime then. What is the showtime? Showtime meaning that changes in the the nucleus divisions. Okay, it's either mitosis or the meiosis process. So basically, when we calculate, so interphase is 98% or approximately 92-97% of the total of the cell cycle itself. Okay, so the 98% will undergo interphase. Okay, so can be divided into three. Okay, first is G1, where the cells grow before they need to duplicate. So basically, from the small cell, they will grow into a normal size of the cell. So normally, when we have the one of the chromatid here, all right, so they are preparing for the duplication process in terms of the DNA thread. All right, so at the end of the uh, uh, G1, they want to go to the synthesis process. Synthesis is a phase where the DNA is a duplicate, meaning that the chromosome is become a double from a single chromatid, they become a sister chromatid. Alright, so when they're reaching to the G2, alright, G2 is period where DNA is being duplicated, where they prepare for their cell division. So basically, before they enter to the prophase, they will have the features of the DNA to look like this one, right? Meaning that they are ready for their showtime, which is a mitosis process. Okay, so when we're talking about prokaryote, since that prokaryote do not have the nucleus, they only have a single ancestral chromosome, which is they only have a simple life. So prokaryote only have a simple cell division through binary fusion only. Okay, so they are not complicated like human. Okay, so if you asking me why they are not complicated, because they have a simple life. All right, so please do not complicate your life. Okay, uh, so prokaryote is a very what we call is a simple creatures created by the gods, so that life is simple. So enjoy. Right. So they are different from the eukaryote. Eukaryote is what we call is a multicellular organism, like human. Okay, we have a lot of tasks to do. We have a lot of function, and obviously we are qualified of in this world. Right. Obviously, we have more sophisticated system. All right. So eukaryote, which is including the human and so the plant. Okay, they must divide their nucleus through the mitosis or the meiosis. Okay, mitosis is for the cell generation, cell growing. Okay, meiosis only for the cell production. We have to remember that. All right, before the nucleus divide, the genetic material will be replicate or duplicate first. Okay, so eukaryote chromosome is a very long thread-like structured DNA, consists of genes and then located in the nucleus. They are only visible of this nucleus during the cell division, as I mentioned earlier. Okay, only 2% from their life cycle can see through the microscope. Okay, so they are made from, the DNA is made from, the cell is made from 27 DNA, 67 protein, and also 6% of RNA. Right, this is a human. We take one example of the human cell. We look inside of the nucleus. So they have the chromosome, okay, and when we unwind this chromosome, they have this DNA, okay, we call it the double helix, okay, so this is what we learned before. They have the adenine, they have the guanine and cytosine, and also the thymine. So we're going to look at the first one, okay, so this is a mitosis process. Okay, so as being learned before. Okay, mitosis consists of four. Okay, PMAT, PAMAT. Okay, you remember P prophase, M meta, anaphase, and also the telophase. Okay, it's the same thing. Okay, so mitosis is actually consists of the similar to the plant and animal. Okay, but plant they lack of centrioles, right? All right, so they got 5 stages, which is including the interphase. Okay, so this is what we call, as I mentioned earlier, the PAMAT process. Okay, so I do believe that you already mastered this one. Okay, so interphase where cell carry the normal life activities. So the process is become chromosome become duplicate, and then when they have an early prophase, so you can see that the chromosome is they're quite clear and visible, look like a chromosome, not like this. Is a very thread-like, you cannot see anything. Okay, so they are visible but still long. Okay, late prophase where you can see that the chromosome is become shorter and condensed. Okay, and metaphase, line up. Okay, the keyword is a line up. Okay, once the line up is a metaphase. Okay, spindle fiber will attach to the kinetochores and bring the metaphase to the what we call the equatorial plane of the cell. So function of this one, so that during the anaphase, once they divide into two, meaning that the chromosome, okay, just now is the butterfly shape, so now they divide into two, meaning that the chromatid, one chromatid is right here, another one we go to this pole, meaning that they are undergo a anaphase stage. Okay, separate and move to each poles. Okay, once they divide, we call it a telophase. So remember, telophase, there might be difference in the animals and plant. For the animal, the cleavage of furrow. For the plant, we call it a plate. Okay, so this is one of the onion under microscope view. Okay, we're going to look at this one during our practical skill. Right, please spot this one. So this is actually the prophase. Alright, number 2, they are metaphase. You can see that they are lined up here. Alright, they line up here. So after that, they will undergo a process of separation. So this is we call is a anaphase. So and then the last part is a telophase. Where are you? Okay, here. This is telophase. But you can see others. Okay, this is a anaphase. This is the metaphase. This is metaphase also. This is the anaphase. Alright, so here is just, this is, I do believe this is a telophase. Right, so the rest of this one, they undergo the interphase, which is, you call is a resting phase, but actually it's not resting. They got a G1, they has S, and so the G2. Alright, actually they're quite busy. Alright. Cytoplasmic division, or known as a cytokinesis, is where from one cell they become two. They're really different in terms of animals. Okay, animal, they have a contraction of microfilaments that form a furrow or cleavage. So and then they will ultimately will pinch out this one into two parts. Alright, so for plant, okay, they have a series of flat vocals made up of Golgi bodies that appear in the middles, so meaning that in the middles of the cell. Then they will dance and call the fragmoplast. Then they will fuse together and ta-da, they become two. Alright, so this is the plant. Okay, so as I mentioned earlier, this is a cleavage of furrow. This is what we call this for plant. Okay, they made from vocals and merge together to become the two individual cells. Right, same thing. Differences between mitosis in plant and animal. Okay, obviously the most obvious is that central, they are absence of the plant. Central are absent in plant, but they're present in the animal. It's not absent, actually, it's being reduced in number. Ester do not form. Okay, animal cell form. Okay, so they are ester is actually related to the centrioles. Cell division involves the formation of the cell plate, which is for animal the cleavage of furrow location, mainly for the meristem. For animal cell, They are occurs throughout our tissues. Alright, so see you next. So we have significant of mitosis. When we're talking about mitosis, produce identical cell. Okay, so what is the importance? What is the significance of this mitosis? When we're talking about mitosis, actually they will provide a genetic stability to our cell or tissue. So meaning that normally, okay, human they have 46. Okay, when we're growing old, when we're growing cell degeneration and so on, okay, we're going to have a stable number of chromosome. Okay, we won't change into a 47, 47, 48, 49 and so on. So meaning that there are mutation there, right? It's not good. Okay, so basically mitosis, okay, thanks because they provide a genetic stability. Obviously for the growth, okay, to increase in the mitosis, so for growing in our multicellular organism. Cell replacement, if any cell are dying, okay, for example our red blood cell, they're only lasting for about 220 days or 3 or 4 months. So after that they will be replaced in our body, so through the mitosis process. Obviously for cell regeneration, okay, certain part of the body, okay, some animal, for example like the starfish, battery, okay, so they will regenerate the whole parts of the body when they have been cut their arms. Okay, so for the asexual reproduction, meaning that for certain animal and plant, this method is a very they are very useful for them. Okay, because the mitosis process, they will form a new organism. Okay, we go to the meiosis process. Okay, meiosis typically they have two nuclear divisions, and they have the cytoplasmic division. Okay, they're being designed or simply refer as meiosis 1 and also the meiosis 2. Alright, so under the prophase 1, they can be divided into five, what we call five phases. Alright, which is liptotin, zygotin, perchitin, diplotin, and also dikinesis. So we have to remember, after they finish the telophase 1, there is no DNA replication, meaning that this continue to the prophase number 2 immediately. Right, so during the prophase 1, they have 5 substages. Okay, we call it a liptotin. Right, remember the meiosis process is they produce non-identical. So that's why during the prophase 1, they have to exchange some of the materials. Okay, during the prophase 1, they have to exchange of the from the what we call the non-sister chromatid, so that different allow will be exchanged and provide towards the end of this process, they will have different genetic from one daughter cell to another daughter cell. Alright, so liptotin is where duplicate chromosomes start to condense. Zygotin is start synapse begins. Okay, perchitin is actually when the synapses consists of the crossing over will be occurs. Diplotin is where the synaptonemal complex disappear and start to visible. And dikinase is they are ready for the metaphase. Method for genetic recombination. Number 1, we have a crossing over. So obviously for the crossing over, we do have a crossing over between the same chromosome, or known as a homolog chromosome. For example, here we have chromosome number 1 and also number 1. They cannot have a crossing over between chromosome number 1 and number 5. Alright, they cannot be. Alright, so at the end of this crossing over, they will produce the non-identical for each of these chromatid. So this is a result of the meiosis. Why meiosis produce non-identical? Some of the question they might asking you why meiosis produce a different. Okay, the answer is here because they do have a crossing over between the homolog chromosome. This is a meiosis 1. Okay, once they finish the meiosis 1, alright, so they already become a haploid. Okay, so under the meiosis number 2, okay, they just copy the haploid to become two, what we call two copies of the cell. So basically, from this, during the meiosis 1, from 2N they become N, and after the meiosis, at the meiosis 1, they just finish, and then okay, they become haploid, and throughout the rest of the meiosis number 2. Right here we have the small pictures of the what is the meiosis 1 and the meiosis 2. Okay, meiosis 1, they have same the PubMed process PMAT, but then they have the Roman here, number 1. Difference from the meiosis 2, they have a Roman 2 Roman here. Process Prophase 1, they undergo a cytokinesis and so on. Alright, so the main difference here is actually during the metaphase 1. Okay, during the metaphase 1, in the meiosis, the homolog chromosome line up. Okay, you can see it's very clear here. They are double what we call line up here. So, and then making that once they undergo anaphase, they will go here, they go here. For example here, okay, we have a meiosis 46 chromosome. 23 pair will be lined up here, 23 will be lined up here. So each of these cell, okay, they will got 23 here, 23 here. Alright, so at the end of this one, at the end of the meiosis 1, actually they already haploid, meaning that they already have a copy of homolog chromosome half from the normal one. So they are 23, they are 23. Alright, then they continue to the meiosis 2 without the DNA replication. So this one, the metaphase 1, they will line up of the as a chromosome only. Okay, remember the metaphase 1 in this one, they line up as a tetrad. Tetrad also known as a what we call also known as a homolog chromosome that makes these changes. Alright, so right now they do have a normal process, anaphase, separate of the chromatid, and they become a telophase, and lastly become a cytokinesis. And towards the end of this one, alright, so they have 4 daughter cells, which is difference in which is known as a non-identical. Alright, so the end of the meiosis. Okay, so significance. What is the importance of the meiosis? Obviously for sexual reproduction, our gametes, our sperm, and also the ovum, they are made from during the process of the meiosis. And this meiosis occurs in our gametes, okay, for the generation. I mean for the to produce a new generation. Genetic variation, obviously, okay. So even though you are being produced from the same parents, but obviously the siblings are quite different. So that is genetic variations, meaning that a new combination of gene, they might be different from one offspring to another offspring. Okay, so genetic variation are brought by the independent assortment and also the crossing over in the prophase 1, as I mentioned earlier. Here is the difference between the mitosis and the meiosis. You can see and you can read by your own. Okay, so alright, so I'm going to the next one. Alright, so the meiosis and the mitosis, obviously you can see a very difference. Okay, in terms of the numbers of the cell being produced, they only have 2. Okay, this one 4, which is non-identical. This is identical. They have 2 phases. Okay, meaning that meiosis 1 and meiosis 2, they only have 1 phases. Okay, and the purpose of the mitosis obviously different compared to the meiosis. So next, comparing the mitosis and meiosis in terms of the genetic materials. Okay, we're going to look at the mitosis first. Okay, mitosis, remember, only produce the identical. Okay, what is genetic material? Meaning that the chromosome. Okay, so you have one chromatid. Okay, and once they undergo, okay, this one we call is the interphase process, G1S and also G2. So when they finish the synthesis process, they become what we call is double the chromosome. Okay, but will not change the number of chromosomes. Obviously, the genetic material only changes. And once they finish the meiosis, they become single back, meaning that the chromatid. Right, in terms of genetic information, meaning number of chromosome. Number still the same. You have 46 at the early, and at the end of this process, you still have 46 chromosomes. Compared to the meiosis process in terms of genetic material, okay, example, we have the chromatid here, okay. Once they undergo the process of the interphase, they got two chromosomes. And then once they finish the meiosis 1, they have one chromosome. And then when they finish the meiosis 2, they have a single or one chromatid. Right? In terms of genetic, the information means that the number of chromosome, okay, number of chromosome, the normal, okay, at the early process they have 46. Okay, once they finish the meiosis 1, they become 23. Okay, so meaning that and continue to the meiosis number 2. So basically we have finished for the chapter 1. Okay, the details of the process and so on will be in our textbooks. Right, so this is the end of the chapter 1. So thank you very much and really hope that you enjoy our lesson and our videos. I recommend you to watch another 2 videos that will being uploaded later. Okay, so we can't wait to see you in the class. Okay, so really hope you enjoy our lessons. Okay, take care and Alhamdulillah. Okay, see you soon. Assalamualaikum warahmatullahi wabarakatuh.